Welcome everyone to the May installment of the IPM Hour. We're now over a year in the works, so that's certainly good. Today, we're going to have Amy Murillo talk about ectoparasites of poultry, and then I'm going to talk about microbes controlling microbes. Amy's going to go first, and she did her undergraduate work in Indiana and then graduate work in North Carolina and California, and all of that has been focused on entomology. She currently heads up the veterinary entomology lab at University of California, Riverside. She's interested in studying ectoparasites of livestock and has developed integrated pest management tools for commercial and backyard poultry producers. And I thought this was kind of interesting because I'm hoping that this is in the talk today. Interested in studying the effects of ectoparasites on animal welfare using on-animal sensors to measure stress levels. So today, Amy is going to talk about ectoparasites of chickens. Take it away, Amy.
Great, thank you so much. There is a bit of a disclaimer. I will be talking about poultry ectoparasites, and it is a lunchtime thing. So apologies in advance. I didn't think that one through. Thank you so much for this opportunity. I thought it might be nice to introduce myself, to the broader California community and Western Coast community, because I am a new assistant professor. I just started in July during all of this craziness. So it's definitely been interesting, and there have been some challenges. And today I'm just hoping to give a brief introduction to some of the things my lab is interested in, at least the poultry side. Being a livestock entomologist, there's definitely other opportunities of livestock in California, but a lot of the work I've done so far and my immediate work is poultry focused. So why chickens? I'll be specifically focusing on egg layers. So these are the birds we raise for egg production. And this is a huge industry in the United States. Over 95 billion eggs are produced annually here in the US. The per capita egg consumption is over 300 eggs per person per year. And this is increasing. So egg demand is not going away anytime soon. California is consistently a top 10 producer and is increasing, especially in areas such as organic egg production. Now, whatever your interests with the poultry industry are, personally, you've probably started to see some of the changes going on. You've seen at the grocery store the increased availability of things like cage-free eggs. This has been all over the news. You might have started to see other options like free range or organic, and may not know what those mean. And these changes to the egg industry, some of them are being driven by consumer demand, and some of them are being driven by legislation. So for example, in the state of California, legislation has recently been passed that will prohibit the use of chickens raised for eggs to be in cages. So this started as an individual, company. Here's some big names I've thrown up here, but this is starting to matriculate into everyone in California having to use this cage-free egg supply. Now, to give you a little bit of background, what does this mean? So this is an example of a conventional caged poultry farm. This is a relatively small one, and I do want to mention that there are a lot of different configurations that a conventional farm could look like. So this is only one tier of cages, but the things I want to point out is that the animals are confined, they're held at high densities, and one of the main reasons they're even in these cages is you'll notice they're up off the ground away from their manure. So this is really a driving factor in putting these birds in the cages, was to reduce disease transmission, because a lot of disease risk for poultry is fecal oral spread. The other advantage to this system is that both the egg collection and the food and water are automated. We do have this intermediate stage. They're known as enriched or furnished cages. In these systems, the birds are still confined. However, there is more space per bird, and they have these added enrichments such as perches and scratch pads to promote good behavior and good welfare. And the birds are, again, mostly separated from their feces in these systems. When eggs are labeled as cage-free, this is what a typical system will look like. The birds are still confined to a structure, so they're usually in large barns, but they're not individually confined, and they have a lot more freedom to move around. Uh, as you can see, they're on the ground, so they are potentially in contact with their feces. And in these systems, egg collection can start to become a little bit tricky. If eggs are labeled as organic, they would be by law cage-free, and these birds would also have to have access to the outdoors as well as have organic feed provided for them. And finally, at the far end of the spectrum from these conventional cages is free-range birds. These birds are outdoors foraging in the most natural environment. And then there's some sort of mobile coop that's often provided for them to seek shelter at night. There are nest boxes provided, and these mobile coops can be moved from pasture to pasture to provide forage for the birds. Now, regardless of the system that the chickens are in, I'm an entomologist. I'm interested in the ectoparasites. And the two most common ectoparasites we see in commercial poultry are the northern fowl mite and the chicken body louse. They both live on the birds, so they don't actually care where the birds are. They are more focused on the host. Now, the northern fowl mite is the most common and damaging ectoparasite in commercial poultry. They are blood feeders, and they prefer to live in the fluffy vent feathers of the chicken. And if you haven't met a chicken, the vent is essentially the butt of the chicken, and they have these super fluffy feathers. And that provides the proper microhabitat or microclimate for these mites to really thrive. Mites can have a very short life cycle. They can go from egg to adult in as few as 7 days. And because of that, they can reach very high populations very quickly. So what I will show here in a moment is a video of a bird that had been infested about 4 weeks prior. And all these little dots on my hands, these are my hands that are being recorded. These are all adult mites. Hopefully this video works. Yeah, there we go. So these are adults from the chicken crawling, and gives you an idea of the size of the mites as well as just the sheer number. Fortunately, these are bird parasites. They're not human parasites. So while they are crawling on me, they're not biting or feeding or infesting me in any way, which I'm sure is very reassuring. Mite economic data impact has been very well studied, at least in cage systems. So mites do decrease egg production through this blood feeding. They can cause anemia and mortality. We also see reduced feed conversion efficiency. So the birds are eating more food to produce the same amount of eggs. And this is very expensive for producers. Feed costs can be one of the most expensive parts of the whole operation. One study conducted over 10 years ago, I'm realizing now my own mortality, showed that profits were reduced 7 to 10 cents per hen in just 10 weeks. And that might not sound like very much, 7 to 10 cents. However, at the time that this study was conducted, the profits for a dozen eggs was only 7 cents. So at the time of this study, the mite infestations were essentially completely reducing the profits from eggs. Chicken body lice have a different life history. The lice are, first of all, they're found all over the body of the birds. They're not limited to one area. They are primarily feather feeding. They're a little bit larger, and lice have not been quite as closely linked to economic data. A lot of the research is a little more mixed in how much damage they cause. However, they are very visually striking on the birds. So excuse me, the feather material, because it's eaten, the birds look very ragged and kind of sad. And then the eggs, which are here shown on the left-hand side, they're laid in clumps. So the birds will have these big clumps of feathers that are heavily laden with eggs. Chicken body lice can feed on blood as well. This is the midgut of the louse, and this dark coloration, this is actually blood. So the lice are opportunistic blood feeders. Traditional control has heavily relied on pesticides. Um, In a cage system such as this, it's relatively easy to go down the rows and to spray the birds directly. These insecticidal materials have to be applied at very high pressures, and they're usually directed underneath and then up onto the birds so that they can get into the feathers, especially where the mites are. Um, However, in recent years, there have been issues with pesticide applications. First of all, there are few available compounds that are actually approved for use on animals or food animals. And because of that, we are seeing lots of insecticide resistance to those few compounds. If those challenges weren't enough, now as we look at cage-free egg production, we can't just spray pesticides the way that the caged layers could be. So the challenges here are not just what materials do we use and if there's resistance to them, it's also how do we even apply insecticidal materials to achieve any sort of mite management or louse management in these types of systems. So one possible solution is to encourage the chickens to treat themselves. Self-treatment does have its advantages. This is less stressful for the birds. If you can imagine trying to run around spraying at the birds, that would be very stressful for them. They would not like that very much. It also can be less labor intensive, depending on what you use. If it's a one-time setup and then the birds do all the work themselves, that could definitely save on time and costs. And it has the potential to work for longer periods of time. So not necessarily that the material itself has longer residual, but that the birds could be treating themselves not just at one place in time, but continuously over the life of that flock. So one possible solution is to take advantage of a behavior that chickens perform naturally, which is called dust bathing. Chickens perform dust bathing, and this is seen in other birds as well. You might have noticed it in wild bird populations, but they will rub themselves into dirt or sand, and they do this to remove excess oils from their feathers and keep their feathers in good condition. So what this video is going to show is a chicken performing the dust bathing behavior. And she's really going to work this material. This is sand. She's really going to work this into her feathers. And if you remember, the mites are on the butt of the chicken. They're in those feathers. So this material is getting worked into those feathers. Now, sand or dirt on its own won't do anything to decrease mite or louse populations. However, if we introduce an additional material, such as diatomaceous earth or sulfur dust to that material, we can achieve good control. And that is exactly what we tested here at UCR. This was part of my PhD work. We introduced diatomaceous earth into sand, and we gave chickens the opportunity to dust bathe in it naturally over a period of time. At one point, so the birds started uninfested. There were no ectoparasites. And then we introduced ectoparasites after the birds had access to these dust boxes. And what we found is that when the birds had access to the dust boxes, there were very low ectoparasite numbers. It wasn't completely zero, but they were kept very low, which would decrease the economic damage that the mites could cause. When we removed those dust boxes and took away the dust, even though there was some still in the environment, we saw mite populations rebound very quickly. So the ability for the birds to actively dust bathe in this insecticidal material achieved very good control. It also achieved good control whether or not the birds, individual birds, were using it regularly. So we did track relative use of the boxes on all the individuals as well as the individual mite levels. And what we found was that we achieved flock level control, which is really ideal and what we would want in a commercial setting. With any good experiment, we replicated it, and then we saw something a little bit different. In our second trial, we had extremely low mite levels when the dust boxes were present. And then we removed those dust boxes and we saw a normal rebound. So the arrows here are indicating when we removed the dust boxes and took away the diatomaceous earth. And what we noticed was that our birds looked a little bit different, even though we had sourced them from the same farm, they were the same breed, same age. We noticed that their beaks were different. So the method that the farm used for the birds that we used in trial 1 was what is called a hot blade beak trim. Beak trimming is a normal practice for commercial birds. This is done when they're very young, and it is to prevent the tip of their beak from forming completely. And this is because the beaks are very sharp, and birds have this natural pecking order, and they can cause damage to themselves or others when their beaks are intact. And so to prevent morbidity or mortality, the beaks are trimmed at an early age. In the second trial, that same farm had switched to what is called an infrared beak trim. And this uses a laser beam instead of a hot blade. And you can see that the beak is more intact. And we were able to test this further, and we were able to show that the beak was the difference. And so having the more intact beak with the infrared beak trim actually allowed the birds to groom themselves better and to remove ectoparasites more effectively. So this is an example of a cultural control method that could be implemented with something like the diatomaceous earth and to achieve mite control. Because again, once we remove those dust boxes, even with the infrared beak trim, we did still see mite levels rebound. So the trim on its own was not sufficient. If we revisit these poultry housing types, there are obvious differences if we look at it from the chicken's perspective. But if we look at it from an insect perspective and an ectoparasite's perspective, these environments are very different. The caged birds are not going to allow a lot of off-host hiding spots. Whereas all the other housing on the right-hand side is going to have lots of nooks and crannies very close to the birds that things could potentially hide out on. And so our hypothesis is that as we transition away from caged layers, we're not just going to have our typical mite and louse problems, but we're also going to increase the diversity of parasite pests that we could potentially have on commercial egg layers. And it's very hard to get onto commercial farms for biosecurity reasons. So instead of surveying farms in the area, we went out and surveyed backyard chicken coops. And these environments, even though obviously the densities are very different, the sheer numbers of birds are very different, these environments are a lot closer to cage-free than, at least from a parasite's perspective. So these birds are going to be on the ground, they're going to be potentially interacting with wild animals in the area, and they are going to have access to the outdoors. So my PhD advisor and I, this is Dr. Brad Mullins, we conducted this backyard chicken survey, and we went out to properties all over Southern California. And we either washed the chickens using a permethrin spray and got all the parasites off, or we, on properties that didn't want us to use chemicals, we literally washed the chickens in a little bit of soapy water. The chickens aren't super thrilled about this method, but it is very effective for getting any parasites off of the animals. And what we found was very, very interesting. So we found our typical northern fowl mite. That's our on-host parasite that we see in commercial flocks, but we also saw the chicken mite or poultry red mite. This is a very common ectoparasite in Europe, where they've had cage-free housing for over a decade now. And these are an off-host parasite, which means that they live in the environment, and then at night, while the birds are sleeping, they come out and they blood feed then. And these images on the right-hand side are from a European commercial farm, and you can just see the sheer numbers of mites. This video I'm going to show on the left is actually from a farm in California. And these are going to be mites that are on the perch, and I'm poking it with a stick because that's what you do with bugs. And all these little writhing things, these are all mites, and they're all very happy and very blood fed because they've been feeding on chickens. We also found stick-tight fleas. So these are not like your typical cat or dog flea where they're just hopping around from animal to animal. These fleas actually embed themselves in the skin of their host. So on chickens, they really prefer the face, the combs and the wattles of the bird. And the adults, both the males and females, will blood feed on the host, and then the eggs will drop into the environment, and the immatures will develop then in the litter near the birds. Stick-tight fleas are not poultry specific. They will feed on lots of different vertebrates. And so many times on these properties, they would have cats, and the cats would be laden with these fleas. They can be on dogs. They're oftentimes on wild rodents or ground squirrels. So these can be rather ubiquitous once you find them. We also found scaly leg mite. This mite is related to the mite that causes mange in cats and dogs, companion animals. But instead of causing the fur to fall off, these mites attack the skin of the feet. And so they can actually cause the scales of the feet to fall off, which can cause some major issues for the birds. And finally, we didn't actually find bed bugs during our survey. However, there have been increasing reports, and people have started to send me bed bugs, which is always fun, from commercial poultry facilities. So these are bed bugs from a commercial poultry facility. They have a very similar life history to those chicken red mites, where they're in the environment, hiding in the perches or nest boxes, and they wait for the chickens to sleep, and then they travel at night to feed on them. These are the same species that impact people, and there is the potential for them to host switch as far as we know. So this is definitely an emerging problem. And here are some images. These are from a poultry facility in upstate New York. So to kind of continue on that thread, this is a project that started just before the pandemic. We're actually attempting to survey commercial poultry facilities in the US using a trapping system. So obviously we're not traveling, but it's also for biosecurity. And we're sending out traps. They're cardboard. There's one shown up here on the top right-hand side. And what they do is they provide harborage. So if you put these traps out, mites or bed bugs can go hide in them. And then when you collect the traps, we process them. This is what we find. So we're attempting to kind of more concretely show these emerging pest problems in commercial poultry, not just using backyard poultry. And if you're interested or think know anyone might be interested, you can find out more information on this website. Finally, just a quick mention, and Matt mentioned this at the beginning, I'm also interested in how ectoparasites interact with their hosts, and specifically it's mites and lice interacting with poultry. And we've been working on quantifying how mites and lice impact the welfare of birds as well as the behavior. And so for the behavior part of this, we're using on-animal sensors. We call them our chicken backpacks, and they have these little sensors in the backpack that pick up the movements of the birds. And we've been working with computer scientists here at UCR, and they have helped us to create an algorithm. Well, they've created the algorithm, we've helped them by providing data. And what we can do is we can track specific behaviors that the chickens perform. So preening or grooming, dust bathing, as well as we're using pecking as a proxy for foraging. And what our results show so far is that northern fowl mites really impact the preening of chickens. And what we hope to do with this information, obviously a commercial farmer could not put a sensor on their tens of thousands of birds. However, we're hoping this information can help us inform monitoring strategies. So what behaviors might you look for in the birds that might indicate that there's a problem and we need to do some more hands-on monitoring and maybe implement a control method? It might give us an opportunity to evaluate control methods. And also, I want to continue doing some of this work and really help to get good economic data from cage-free systems and really try to identify how these ectoparasites might be impacting the industry as a whole. With that, I've got lots of people to thank. This is a collaborative effort. I'd like to thank my funding resources. And obviously, we're just getting started here at UCR, but we're really excited and yeah, looking for collaborators. So thank you so much, and I'll take your questions.
Thanks, Amy. That wasn't nearly as bad as I thought it was going to be in terms of imagery. Although the mites crawling around on your skin was disconcerting to say the least.
It's weird that that becomes normal.
Um,
Okay.
yeah, so you want to go ahead and read the question?
Yeah, so question in the chat, what are the typical cage-free system sanitation practices between sequential laying flocks? That is a great question. And I don't know if there's a standard. Laying hens are usually kept for about 2 years before they're depopulated. And broilers are there's a lot more frequent turnover. So I'm not sure how much downtime there is between flocks. Um, I don't know that there's anything typical I can tell you. I'm sorry. I should
Cool.
mention that a lot of the mites that live off the host, they can survive up to 9 months between flocks without a blood meal. And I know they're not leaving them down that long. And that's something that's really been targeted in European flocks where chicken mites are a major problem, is trying to control the mites in between flocks and using strategies like heating. But it doesn't take very many mites to survive to reinfest the new flock. And that's really one of the biggest challenges with ectoparasites like mites, that they're just really, really good at reproduction. Are there any IGRs used in this system? I'm not aware of any IGRs that work for mites. There are some IGRs that will work. There are other pests I didn't even talk about, like litter beetles, that have been targeted for things like bed bugs. IGRs, they do work to a certain extent, but even the immatures are blood feeding. So as far as like a holistic control method, they're not always the best for things that are going to continue to be pestiferous in the immature stage. Would the self-treatment dustbins work on a commercial scale? That is a great question, and that is something I want to test further. We have been using it as a technique for lots of backyard farmers. It works very well in a backyard scale and small commercial. There are definitely some things that we need to test on a commercial scale, such as like how many birds per bin, how frequently it would have to be refreshed. That's something that we have not yet had the ability to test, but I'm hoping that's one of my goals for sure, is to find a partner. In relation to biosecurity, are other birds at risk for introducing ectoparasites? I would say yes. I think a lot of these the mites for sure are not chicken specific. They can be found in wild bird nests. All of the parasites I mentioned are cosmopolitan, so they're found all over the world. The lice are a little more perplexing. We're not really sure where the lice are coming from, just because lice tend to be more, not just chicken specific, they can be on like turkeys and pheasants and related galliforms, but the passerine sparrows or pigeons that are more ubiquitous, we're not really sure if lice are accidentally on them short term to move them around, or if some sort of I don't know if you guys know, louse flies might be moving things. Sometimes they're keds, they're a blood feeding fly. They can have lice, uh, use them for foracy and move around. It would be very good to survey some of these areas around poultry farms and see if we can find spread either out or in from these facilities that are infested. Another question, crossover of poultry parasites to pigs, cattle, goats on small farms. The lice probably aren't moving around, like I said, unless there's like pheasants or turkeys, but the fleas could infest pigs, cattle, or goats. The mites are probably still going to be bird. They are really bird specific. So there's probably not a risk to the other animals that way. And there probably aren't any parasites moving from those animals to chickens either. So they're all going to have flies, which is, our number one pest, right?
All right. I'm not seeing any more questions in the chat.
And I can
Are there?
continue to answer those if they pop up.
Okay. Okay. So let's go ahead and switch gears. I unfortunately don't have any videos of mites crawling on people's skin. Maybe that's a good thing. Steve, you want me to just go ahead and introduce myself?
You're our introducing guy, so yes, you have to.
I guess that's true. So should I refer to myself in the first person or the third person is
Do
the
not
question.
refer to yourself in the third person.
Okay. I will not refer to myself. So I did my undergraduate work here in California and then did graduate work in Kentucky where I studied foraging ecology of parasitic Hymenoptera. I currently serve as the acting director for the Western IPM Center, and that was all I was going to give in the way of introduction. Let's see. Now I just need to change this to. Oh man, I can't see it. Great. All right. So today I'm going to talk about ecological interactions that occur among microbes in soil communities. I'm not really going to talk about interactions that occur between microbes and some of the larger organisms like plants and insects. And most of the examples that I use in this talk come from biological control agents that have been used in agriculture. The specific type of biological control I'm talking about here is really probably best classified as inundative. And in this case, what we do is we inundate the system with the biological control organism and then reapply as necessary. So significant interest exists in the biologicals market, and this may be related to the growth in the organic sector over the past several decades. About 30% of the biologicals market is devoted to products that include the microbes themselves. The rest of that market is compounds that are derived from the microbes or from plants, and I'm not going to be discussing any of that today. So really just 30% of that overall market. Now, I did want to take the opportunity here to talk a little bit about the discovery part of these microbial products. These microbial taxa that have been used primarily in these products have been identified as being associated with disease suppressive soils. So these studies generally show a correlation between the presence or absence of certain microbes and the suppressiveness of plant pathogens. But we can break out this suppressiveness designation into two categories, general and specific. In the case of general factors that that cause a disease suppressiveness, these are oftentimes microbial communities that are involved in antagonism with the plant pathogens. Specific factors or mechanisms are specific microbes that affect the plant pathogens in very specific ways. So this might be antibiosis or parasitism. And importantly here, it's been suggested that these specific factors might be more useful, might be more important for inundative biological control efforts, primarily because they're transferable. We can pull them out of the system and put them into a different system and still have them be effective. Currently, much of our thinking about pesticide products is in terms of modes or sites of action. For our discussion here, I'm only going to focus on the FRAC groups. So this is the Fungicide Resistance Action Committee groupings. And there are three in particular that I'm interested in, B, M, F, and P. The BM group covers microbes with modes of action that I guess would probably be best characterized as ecological in nature and relate to species interactions, things like anabiosis, competition, and parasitism. In this talk, as we will see later, I'll actually break up competition into direct competition and indirect competition. Indirect competition is resource or substrate competition, and direct competition might be considered as antibiosis. The F grouping covers products that affect cell membranes, either their synthesis or their integrity. It should be pointed out here that these cell membrane disruptors probably play a role in competition or parasitism among these microbes. And the last category, P, relates to the induction of host plant resistance genes through the colonization of root zones by the applied microbes and then signaling between the plant and the microbe. I'm not going to talk about this today because this is a pretty complex subject and I don't have enough time. But that's not to say that this is not an important component of biological control using microbes. I just don't have the time to do it today. So as I mentioned before, this is primarily a literature review. So I went through the biological and agricultural databases, including Web of Science, Agricola, and Google Scholar, using the terms that I got from the FRAC modes of action, as well as ecological terms describing interactions among microbes. Most of the literature is about 10 to 20 years old, and many of these are review articles that are in the primary literature, although I did look at some primary research articles. So single-celled organisms have been around for a really long time, about 3.5 billion years. And during this time, microbes have evolved symbioses that among the various species in the microbial community. And there's also been ample time for metabolic diversification and specialization. And one specific aspect I wanted to talk about here is the endosymbiotic theory for the evolution of eukaryotic cells. And the reason I want to talk about it is because, as we'll see in talking about this theory, that in fact those interspecies, those interactions among species can change depending upon context. So according to the endosymbiotic theory, eukaryotic cells arose when free-living bacteria were taken inside another bacterial cell as an endosymbiont. So mitochondria probably evolved from proteobacteria, and chloroplasts probably evolved from cyanobacteria. And it has been suggested that eukaryotic cells may have evolved from predatory bacteria like Del Vibrio that may have penetrated into the larger bacteria to later become an endosymbiont. These type of symbiotic relationships among microbes are common even today, and it's common that cells that have been engulfed by another microbe don't wind up being consumed, as was the original intention probably, but in fact become an endosymbiont within that host cell. It's also important to point out that single-cell organisms like bacteria can function in unison and under high degrees of organization. Early prokaryotes probably developed the means for interacting among themselves and with other groups in a manner that was allowed them to survive. Cell-to-cell adhesion and signaling are two mechanisms that are widespread in the bacterial world. For instance, Myxococcus, which we'll talk about in a little bit, when starved of nutrients, produce signals that cause aggregation and gliding along chemical gradients. And in fact, it also has been suggested that the interactions among microbes within biofilms might be considered some degree of symbiosis. So these interactions can be kind of classified along a continuum. And so what I've done here is I've kind of graphed out, if you will, this continuum with transitory with minor impact on the left, and then long-lasting dependence or endosymbiosis on the right. And what you could see is that niche overlap as we move from left to right increases. This is also shown by the two dots above the niche overlap sign, where the yellow dot and the white dot represent the two species in the two different niches, with the left-hand side being basically no niche overlap, the right-hand side being almost complete overlap, and also the strength of association between the species in this interaction. So I'm not really going to discuss the transitory impacts much more, because we're really not going to talk about that, because it doesn't seem to be all that important. And we're going to focus mostly on competition, predation, and parasitism. So competition is a fundamental component of the microbial community ecology, and is thought to be responsible for the assembly, diversity, coexistence, functioning, and evolution of microbial communities. Microorganisms directly antagonize each other via antimicrobials or compete for shared resources. These two forms of competition, direct and indirect, may not be mutually exclusive, and in fact may function in concert to direct an overall competitive interaction. Moving from left to right, predation has been described in actinobacteria like Streptomyces, and in the proteobacteria like Myxococcus. In the case of predation, of course, the predator winds up killing and consuming the prey, thereby reducing the prey fitness to zero. Although predation is not listed in the FRAC modes of action specifically, it is pretty well described in the ecological literature and in a great deal of depth, so I thought I would cover it here. Predation does appear to be facultative in some cases, and it's not immediately clear, but it does seem like it may function in a density-dependent manner, which might be important for biological control considerations. Parasitism of fungi by other fungi, so mycoparasitism, has been very clearly described in the literature, and the example that we will talk about below, trichoderma, is a well-described mycoparasite. Parasitism of fungi by bacteria does occur, but I wasn't really able to find any specific examples from microbial controls, so microbes that were being used in biological control efforts. So I'm not really going to cover that. And then, of course, endosymbiosis, I'm not really going to talk a lot about here. I just wanted to point out that actually the proteobacteria Burkholderia is available as a biological control organism, and this particular example with Rhizopus, which is a fungus, that Burkholderia species has become an endosymbiont. I couldn't find any real example or indication that they had started off as a parasitic relationship, so I'm not really sure if that's true. All right, so let's just talk about these specific interactions then, and we'll start with direct competition or antibiosis. So Bacillus species are probably the best example of this, and this includes a variety of different species like Amyloliquefaciens, Subtilis, Cereus, Mycoides, and others. And all these species are known to be efficient producers of antibiotic molecules, and in fact, some of these species have as much as 10% of their genome devoted to the synthesis of polyketides, like the antibiotics that we normally see. These specific factors, so in this case, what we're talking about is the specific factors or specific suppressive soil type factor. These specific factors are most likely to work in an inundative biological control manner because you can isolate the microbe, you can rear it up in large vat fermenters, and then reapply them. But it's interesting to note that when I looked for evidence that direct competition of this sort was related to success of biological control, I wasn't really able to find a lot of information. Of the two studies that did address this point specifically, one by Jerzy Nowak and colleagues and the other one by Monica Hofty, they felt that indirect competition or resource competition might be more effective at suppressing plant pathogenic targets. because taxa like Bacillus are really good at producing these secondary metabolites, there have been attempts to correlate secondary metabolite production and competitive ability or ability to exclude plant pathogens. So there do appear to be links between the phylogeny of some Bacillus species and secretory systems and secondary metabolites that may help those species cope with specific conditions and adapt to a particular niche. But at least for these Bacillus species that I'm showing here, those secondary metabolites are produced across all of them. Most of those secondary metabolites are broadly expressed across all the different species. So there didn't seem to be any real relationship, as far as I could tell, between the production of these polyketides and other secondary metabolites and competitive ability. So when we talk about resource competition or exploitation competition, what really becomes important is the concept of niche. The ecological niche means the function of a species in a community. And of course, each bacteria or fungus has particular nutritional requirements and biochemical abilities and degrees of tolerance to environmental conditions that help to define where they occur and that niche. The ability of a microbe in fulfilling its function in a particular ecosystem is determined by basically all the normal physical, chemical, and biological factors that you would think would affect that niche. So we're not really talking about all niches here that occur in the soil. We're really more focused on one, and that's the niche surrounding the root zone or the root zone niche. And if we want our applied microbes to be effective biological control agents, they must be able to colonize and proliferate in the presence of existing microbes, including pathogens, in this preferred niche. And it would appear that this applied microbe, the one that we applied as a biocontrol agent, would have to be able to competitively exclude the pathogen for it to be effective. So, it's important to think about how the microbe's niche might be aligned with this with this rhizosphere or root zone when you start to think about how competitive exclusion through resource competition might occur. So, of course, these bacteria that we apply or the fungi that we apply have to be able to get to that root zone. Obviously, we're not applying them directly, or even if they have to find other sources of nutrients, they have to be able to move through the soils. And so chemotaxis winds up becoming an important part of bacterial ecology. So I thought I'd take a moment to at least talk about it here. So chemotaxis is widespread in mobile microbes and obviously is important. Usually the chemotactic response mechanism results in the accumulation of microbes in preferred areas and sort of their absence in areas that are not preferred. For bacteria, of course, here what we're talking about chemotaxis, it's just the movement towards or away from a chemical cue or signal. For individual bacteria, it kind of looks like this. In the absence of any stimulus, and this is the pane that's to the left, right next to the term chemotaxis, you could see that it's homogeneously blue, so no chemical stimulus or no chemical gradient there. And then to the right, that pane shows kind of a purple chemical stimulus, which that bacterium is responding to. But in the absence of any stimulus, a bacterium like Bacillus subtilis swims in a smooth straight line for several seconds, referred to as a run, and then it thrashes around for a fraction of a second, called a tumble, and continues to move in a new and random direction. However, in the presence of a stimulus, and so what these bacteria do is they tumble less often when the stimulant is increasing and start to tumble more when the stimulus starts to decrease. The bacterial flagellar motor, or BFM, is a rotary molecule that's embedded in the cell membrane of numerous bacteria like Bacillus subtilis. And it turns that flagellum at the back end of this bacterium kind of like a propeller and causes movement. Now, of course, not all bacteria that respond to chemical cues have these flagellum rotors. In the case of myxobacteria or myxococcus, they actually use these small pili that wind up pulling the cells forward. Right, so predation. So in the case of predation, the predator has to kill and consume the prey, right? And so this, again, reduces the prey fitness to 0. That's a characteristic of predation. There's often a size differential between predator and prey, with the predator being larger, if you think of owls consuming mice. But that size differential can shift when predators hunt in a pack. So think of wolves hunting a bison. On the microscopic scale, protozoa and bacteria also consume prey organisms. Predation is found again in the actinobacteria like Streptomyces and in the protobacteria like Lysobacter and Myxobacteria. In the case of some predatory microbes, predation is facultative and may be observed under nutrient-poor conditions. The specific example that we're going to talk about today is Myxococcus xanthus. This is a generalist predator and able to feed on many different bacteria and some fungi. And it is likely that different mechanisms are involved in finding and consuming those different prey items. So the predation process involves encountering and recognizing the prey, lysing the prey cells, and then metabolizing the cell contents. Excuse me. Myxococcus finds its prey by gliding over surfaces, either individually or in large clusters, embedded in polysaccharides or sugars. There is probably some sort of specialized chemosensory system that allows Myxococcus to locate its prey. Once it's close to its prey, Myxococcus changes its behavior by stopping and repeatedly reversing direction. These cell reversals can be seen, and you can find YouTube videos of this, as a characteristic rippling pattern as the Myxococcus cells synchronize their motility and reverse in oscillating waves. Predation factors that cause lysis of the prey cells are those that you would typically see in bacteria. So these would be peptidases, proteases, chitinases, glucanases, those sorts of things. There hasn't really been, as far as I could tell, any study that identified specific lytic factors or other antibiotic factors that were involved in predation. So it seems to be generally thought of that most of these are involved at some level. So parasitism is a symbiotic relationship between species where the parasite lives on or inside the host for possibly an extended period of time. For the discussion here, we'll simply specifically address one species, one genus, Trichoderma, which is a mycoparasite. So there was some confusion in the literature about the ability of Trichoderma species to both function as mycoparasites and also as saprophytes. And, it's a little hard to tease these two things apart. It seems like it should be pretty simple, but it turns out like it might be kind of difficult. So it's not really clear how much, if this parasitism is facultative or not. Trichoderma parasitism involves host signal recognition and chemotropic growth of mycelium toward the host. Following direct contact with the host hyphae, Trichoderma coil their hyphae around the host fungus and start aprosoria formation. So you can see the aprosoria in this image here on the slide. Those are two spores with hyphae growing out. And at the end of two of the hyphae, one from each spore, you see this thickened area, and that's the aprosoria. The direct contact is mediated by cell surface lectins and proteins, both on the host and the mycoparasite. And when those signals are turned on, you see a signal cascade, which then changes protein expression in Trichoderma. the Aprosoria starts to produce lytic enzymes and polyketides, or antibiotics, that function together to weaken the host cell wall and allow for penetration of the host hyphae, followed by the attack on cellular machinery with the help of these typical lytic enzymes like glucanases, chitinases, and proteases. Now, interestingly, in a review of density-dependent parasitism of soil-borne diseases, Bruce Jaffe concluded that the parasitism by trichoderma of fungal pathogens probably operates in a density-dependent manner. Right, so I'm just going to shift gears one more time. This is the last slide that I have, but it's a little complicated. As I was thinking about how we might be able to think about these different interactions and the likelihood of success of the biological control organisms, I went to look into the ecological literature to see if there might be some theoretical construct or some theory that might help me understand the likelihood of success. And it seems, at least in the literature that I reviewed, that competition, either direct or indirect, was considered by most as being an important factor in the soil suppressiveness of plant pathogens. So this caused me to look primarily to this sort of theory that was advanced back in the mid-2000s by Peter Chesson on coexistence, or coexistence theory. So the idea here is that we might be able to predict the competitive exclusion or coexistence of two species using modifications of the classic Lotka-Volterra competition model, one of the things that we probably all saw in community ecology. For me, that's quite a few years ago. In this model, what the two most important considerations are niche overlap and average fitness between the populations of the two species. So how does this work? How would this work? So if a species A is consuming resource A, it has a negative density-dependent negative feedback loop with the resource. As it consumes the resource becomes less available and it becomes more limited by it. And we have a second species B that consumes a resource B. Also, as it consumes that resource, it becomes more limited by it. So in this case, there is limited or no resource overlap, and each species depresses its own growth more than it depresses the growth of the other species. And if we assume that those two resources are equally rich in terms of allowing the microbes to grow, what we would expect to see is coexistence of those two species. However, if we change the situation slightly, we still have no niche overlap, but we change the relative or the average fitness of those two populations by changing the resource, then we see an entirely different outcome. So for instance, if one resource is richer than the other, which then leads to higher fitness associated with the species feeding on that resource, if the difference in average fitness between those two, A and B, is big enough, we would expect to see competitive exclusion. If we think about this in terms of what we've discussed today, we might think about the likelihood of competitive exclusion in the case of microbes that are expressing these molecules that are related to competitive ability. Remember, we all assume that antimicrobials or antibiotics are related to competitive ability, but there may be a trade-off between the competitive ability of these organisms based on the fitness costs associated with the production of these microbes. So if you think about, say, two microbes that are interacting, one produces an antibiotic which is effective against the other, which would obviously reduce the competitive ability, or would reduce the population of the second, the antimicrobial producing one or species. You would say, okay, yeah, you would expect to see competitive exclusion. But depending upon the fitness cost associated with the production of that antimicrobial, if that fitness cost is not great enough or is great enough, then in fact you might not see competitive exclusion. So this is still a work in progress, and I have some work still to do to see if this theoretical construct might be useful for us, but it is a work in progress. So that is all I have today. Oh, sorry, I went a little bit over. I will be glad to address any questions if I can.
You've answered everybody's questions, apparently.
Okay. Well, I think everybody's ready to go eat lunch. I don't blame you.
All right. I don't see
All
any
right.
questions.
Outstanding. Okay, so I just wanted to remind everyone that our next installment is whoa, what happened there? Oops. Oh man.
It's June 9th.
Is June 9th. Sorry, I hit the wrong button. Is June 9th. So I don't. Do we have speakers for June
We don't have
9th yet?
speakers for that one. Carrie Wimble Rojas will talk about educating urban audiences on pesticides, and Paul Jepson will talk about risk communication.
Outstanding, outstanding. So thank you all for coming today, and we will see you next month.